started. So welcome everyone. Thank you for joining us today for a session of the Winter Lunch and Learn series brought to you by the External Committee at the Graduate College. My name is Susie and I'll be the moderator for this afternoon session. We're very excited for two of our scholars to have the opportunity to share their presentations with you today. And how this session will go is we'll start off with a quick opening remark, and then I'll introduce our presenters. Each of our presenters will have about 20 minutes to share their presentation with us, and at the very end, there'll be a Q&A period. Please use the Q&A box to enter your questions at any time during the presentations. You don't have to wait until the very end to ask. And finally, we'll conclude our session with a quick summary and concluding remarks. Now, we would like to take this opportunity to acknowledge the traditional territories of the people of the Treaty 7 region in southern Alberta, which includes the Blackfoot Confederacy comprising the Siksika, Piikani, and Kinase First Nations, as well as the Tsuut'ina First Nation and the Stoney Nakoda, including the Chiniki, Bearspaw, and Wesley First Nations. I would also like to note that although we are connecting virtually today, the University of Calgary is situated on land adjacent to where the Bow River meets the Elbow River, and that the traditional Blackfoot name of this place is Mokinstisi, which we now call the City of Calgary. And City of Calgary is also home to the Métis Nation of Alberta, Region 3. A quick description of what our Learn to Learn series is. It is a public presentation series that aims to bridge the graduate college with the Calgary community. So during each of the Learn to Learn presentation, graduate scholars present their diverse research for the public. Our series has been ongoing since April, so today is our 1-year anniversary for this event, and we're continuing the series into the spring. So without further ado, I'll go ahead and introduce our two speakers today. So first up, we have William Nguyen, who is a master's student in the Department of Physiology and Pharmacology. His presentation today will focus on the effect of focal adhesion kinase inhibition in inflammatory bowel disease. And next up, we have Najratun Pinky, who will be giving us a presentation today on multimodal neuroimaging biomarker for sport-related concussion in youth. So now I'd like to open up the floor to William. William, please go ahead and share your screen, and you can go ahead.
All right. Is this good? Can everyone see it? All right. So thank you everyone for attending today's April Lunch and Learn. So yeah, like Susie mentioned, I'll be talking about the effect of focal adhesion kinase inhibition in IBD. So IBD describes disorders that involve the chronic inflammation of the digestive tract. The two main manifestations of IBD are Crohn's disease, which can affect the entire GI tract, and ulcerative colitis, which affects exclusively the colon. Both are chronic relapsing disorders with complex and not entirely well understood mechanisms of development, and is characterized by symptoms including diarrhea, rectal bleeding, abdominal pain, fatigue, weight loss, all of which make this disease highly problematic and painful. Furthermore, it is estimated that approximately 270,000 Canadians and 3 million Americans suffer from IBD, with the annual direct and indirect medical costs in the range of billions of dollars. As such, the rise of IBD also comes with a growing socioeconomic burden. So the cells I'm interested in are eosinophils, and these were first described in 1879, and their high affinity for dyes allowed researchers to visualize these cells in a range of tissues and develop hypotheses based on these early observations. So they are one of 4 types of granulocytes, which are a type of white blood cell that has small granules containing chemical mediators, used in order to combat pathogens. Some of these chemical mediators include major basic protein and eosinophil peroxidase, which are toxic to both the pathogen as well as the host. Traditionally, eosinophils have been associated with allergic and parasitic inflammation. However, more recently, they have also been shown to have roles in diverse processes, including the development and intestinal health. The accumulation of these cells in the GI tract is a common feature across numerous GI disorders, including inflammatory bowel disease. So a pivotal development in eosinophil research was the creation of EOCRE mice. So using the CRE/LoxP system, which is a DNA modification technology used to carry out deletions, insertions, translocations, and inversions at specific sites of the DNA in cells, Doyle and his colleagues inserted CRE recombinase into the EBX gene, which was one of those chemical mediators released by these cells. This has resulted in eosinophil-specific reporter mice expressing fluorescent markers such as EGFP, which is green, and TD tomato, which is orange/red. This allowed researchers to visualize and characterize eosinophils in real time. So under normal conditions, eosinophils make up 1 to 3% of the circulating white blood cells that are present in the colon of healthy individuals. However, during IBD, eosinophil counts can change, and determining whether or not eosinophils are detrimental or beneficial to the resolution of IBD is quite controversial. So in ulcerative colitis, some consider eosinophils to be damaging because eosinophil number and the disease scores often parallel one another. On the other hand, eosinophils are normally present at high numbers in the colonic mucosa, and some data suggest that they may be reparative. And on the right here are just some images I've taken from a mouse colon, which show the eosinophils in those bright orange dots and shapes. So the protein I'm interested in is focal adhesion kinase, or FAK for short. So this protein is involved in cellular adhesion, so how cells stick together and to their surroundings, as well as their motility, so how the cells move around. It is expressed in most tissues, and its sequence is highly conserved across many different species. Attempts to knock out its expression has resulted in distinctive irregular vasculature, hemorrhaging, and worst of all, embryonic lethality. So this is something you simply can't live without. FAK overexpression is associated with a number of human diseases, with many lines of investigation showing support for its role in the development of human cancer. As such, there's been a number of FAK inhibitors which have been developed for the treatment of cancer, and has been shown that treating breast cancer cells with FAK inhibitors have made them become less metastatic due to their decreased mobility. So FAK is a 125 kilodalton protein and is composed of multiple distinct domains. So FAK's activity relies on the phosphorylation, which is the transfer of a phosphate onto specific tyrosine residues. So those residues are indicated by the stars there at the bottom, Y397, 861, and 925. And these are phosphorylated in response to interactions with a number of other proteins. Now, FAK-related non-kinase, or FRANK for short, is nested within the FAK gene itself, but it is expressed independently of FAK. FRANK has been shown to act as a competitor for FAK binding partners, and it has been shown that FRANK expression reduces FAK phosphorylation and disrupts the formation of FAK complexes. And for this reason, FRANK has been applied on a wide range of cells to observe the impact on FAK. So recently, our lab has identified a pathway that prevents eosinophil recruitment in vitro and in vivo in response to IL-4. So IL-4 is a pro-inflammatory molecule. And in this pathway, inhibiting FAK, either with a pharmacological inhibitor or by expressing FRANK, blocks two proteins, BCAM1 and CCL26. So these two other proteins are required for eosinophil recruitment. This ultimately prevents eosinophils from sticking to and migrating to tissues. So this figure at the bottom, on the y-axis are the conditions, and with the two rows at the bottom being PF573, which is a FAC inhibitor, and FRANK. So when it's unstimulated, we don't see a lot of expression, which makes sense. But when activating the cells with IL-4 without the inhibitors, you see that in the media and DMSO, there's a lot of VCAM1, CCL26, and eosinophil transmigration. But once FRANK or the FAC inhibitor is added, we see a decrease in the VCAM1 expression, as well as at the CCL26, and ultimately less eosinophil transmigration. So for this study, my hypothesis is that FAC inhibition can regulate eosinophil recruitment in colonic inflammation. This is split into three aims, with the first one being to quantify and characterize eosinophil recruitment over time in a TNBS-induced colitis model. So for this aim, we used EOCRE mice, in which their eosinophils appear red. And these mice are given enemas of saline or trinitrobenzene sulfonic acid, which is the TNBS, which is a chemical used to induce symptoms of colitis in mice, similar to what is observed in humans. And afterwards, they are euthanized at days 1, 3, 7, and 14, and the colon tissue collected, then sectioned. So to analyze these sections, first I take many, many small images and stitch them together to capture the entire colon section. Next, I subtract the surrounding background tissue in order to visualize the eosinophils alone, and further adjustments can be made in brightness and contrast. And finally, I threshold the image, which isolates the eosinophils even further and helps automate the counting process. So in this figure, the top row shows the control treatments, and the bottom row are sections from mice with TNBS. So these are just zoomed in sections of the colon. And meanwhile, the columns you can see indicates the days that they were euthanized. Meanwhile, on the x-axis, there's on the other figure, on the y-axis, there's the eosinophils in density per millimeter squared, with the x-axis indicating the day. So here you can see that eosinophils peak at day 3 and subside to near control levels by day 7, with the TNBS treated animals showing much greater eosinophil counts. Now looking closely at the images taken from animals on day 3, which was the peak of inflammation, you also notice changes in the morphology and distribution of the eosinophils as they release chemical mediators into the surrounding tissue. So instead of being round and more evenly distributed, they seem to be more clustered as well as more elongated. We also saw a decrease in body weight at day 3. So this figure on the left shows the body weight, the change in body weight on the y-axis, and the days at the bottom on the x-axis. And we see there's a sharp decrease in weight in the black line, which indicates the TNBS animals. We also saw that there's a decrease in macroscopic damage over time. So these damage scores are given based on a number of different metrics, including how bloody, swollen, or thick the colon tissue is, with higher scores indicating greater damage. Next, I also went ahead and examined if there were any differences between the proximal and distal colon. And interestingly, more eosinophils are seen in the proximal colon, which is further along inside the digestive tract. So both areas follow a similar trend, peaking at day 3, although there were many more eosinophils noticed in the proximal colon. To investigate even further, I examined eosinophil counts between the 3 tissue layers of the colon, which are the muscle, the submucosa, and mucosa. And I found that although the eosinophils increase in all layers of the colon, most were found within the submucosa. So I followed this up with aim 2, which was to examine the effect of FAC inhibitors on eosinophil recruitment in vitro following stimulation with pro-inflammatory molecules. So to do this, I grew human umbilical vein endothelial cells and activated them with histamine, which is a compound responsible for many allergic reactions. I followed this up by exposing these cells to varying concentrations of FAC inhibitors, and I can detect whether or not FAC was activated or phosphorylated using a test called a Western blot, which detects proteins based on their weight. So far, our lab has used the following FAC inhibitors. So for the purpose of my study, I was testing FAC14 and GSK225. As for PF573, which we've been using that inhibitor for quite some time in the past. So here's an image of what a Western blot looks like. So on the left-hand side, you'll see what's called a molecular ladder. And since a Western blot separates proteins based on weight, you notice that the phosphorylated FAC band is at the around 125 kilodalton mark. So going through the lanes, the lane A, column A, is the control, B is with the FAC inhibitor, C is adding the histamine or the pro-inflammatory agent, and D, E, and F are where I have the histamine and decreasing concentrations of the inhibitor. So you notice as decreasing the concentration of FAC14, the band gets darker, indicating that FAC was phosphorylated and activated. So more trials need to be done in order to determine the effective dosage of FAC14 and GSK225. But due to COVID and the time constraints, we decided to move forward with PF573. This leads me to my third aim, which was to determine the effects of inhibiting FAC activity on eosinophil recruitment in a TNBS-induced colitis model. So similar to aim 1, I used the EOCRE TD tomato mice, and this time I only looked at day 3, which was the peak of inflammation, and administered 3 different concentrations of PF573, 50, 80, and 125 mg per kg. And again, they were euthanized and their colon tissue collected and sectioned. So we found that there was an overall decrease in eosinophil recruitment in a dose-dependent manner, along with the change in eosinophil morphology and distribution. So similar to the previous figures, this shows eosinophil density per millimeter squared, and we can see that by adding more FAC inhibitor, it decreases the number of eosinophils down to near control levels at 125 mg per kg. So when examining the 3 tissue layers, I saw also the similar dose-dependent decrease with the majority of the eosinophils again found in the submucosa. Interestingly, there was no significant changes in body weight. So we see that for the TNBS, along with all the inhibitors, the change in body weight decreases along the same lengths, with adding the inhibitor not increasing it at all. Along the same lines, we also saw that there was no significant change in the damage scores when the inhibitor was introduced. So to conclude, the quantification of eosinophil recruitment in ulcerative colitis has not been examined to the same degree before, so no one's really compared the proximal and distal or looked at the 3 different layers of the colon. And as such, this project has generated new knowledge about the role of FAC, frank, and eosinophils in colonic inflammation, and specifically a model of ulcerative colitis. So ultimately, these data have the potential to shape therapeutic strategies targeting FAC, either using a frank inhibitor, either using frank or a FAC inhibitor as an agent. So I'd like to thank my lab, the Patel lab, my supervisor, Dr. Patel, along with the lab techs, Evelyn and Hong, as well as the Sharkey lab, my supervisory committee, and the live cell imaging resource lab. And that's the end of my presentation. Thanks for listening.
All right, thank you, William, for the great presentation. So I have a question submitted. The first one asks, how does this potential treatment improve upon today's current treatment?
So good question. The current treatment today involves many different things. So many different anti-inflammatory drugs or immune suppressors, which come with some severe side effects, as well as you can get over-the-counter meds such as anti-diarrheal meds. In some severe cases, people have surgery to remove certain parts of the GI tract. In this case, a FAC inhibitor would be targeting proteins made by the immune system and not alleviating specific symptoms. It might be a good way for patients who are unresponsive to these other treatments. But yeah, that's what I'll say.
Thank you. And now another question. This has two questions actually, but the first one asks, no, it's the same question. So the second question they ask is, why are the eosinophils concentrated in the submucosa?
So correct, I might be wrong about this, but the submucosa is more vascularized and contains many different blood vessels where eosinophils and immune cells can travel, which may be why we see so many of them there as they're migrating towards the tissue itself.
Sounds great. And Stephanie asks, is the immune system between the mice and humans similar?
In many respects, they're similar, which is why they're a good model for so many different diseases. Of course, there's also always the exception, which I can't think of off the top of my head. But mice are used a lot just because they're small and they can rapidly reproduce and they're easy to manipulate genetically.
Thank you. And last question we have here, William, why did you use those concentrations of inhibitors that you have used?
So we chose those inhibitors just based on previous studies. I know one study used it at 80 mg per kg, and so choosing a concentration slightly less and slightly higher gives you a better idea of how the drug would impact the animal.
Thank you. That seems to be all the questions we have. If anybody has last minute questions, feel free to type them in. Otherwise, we will move on to our next speaker. So thank you so much, William, for the great presentation today and the questions answers.
Thanks for having me.
Sounds good. So next we have Pinky. So I'll go ahead and stop screen share. Pinky, please go ahead. Thank you, Pinky. That was a great presentation. So now we'll see if there's any questions. So Nadiha asks, what are the next steps for your research?
Okay, that's actually a very good question because that was not in the presentation. And I intentionally did not put in the presentation because we are still in the process. So initially, when I started my PhD, our idea was to combine these different modalities, because when we do a master's or PhD, people actually normally works with 1 or 2 modalities, but we have like 4 different modalities. So our idea was to, okay, we have great project and we'll put all of them together in a machine learning, and we will like develop a classification model, like who is concussed, who is recovered, and who is controlled, can we classify? But then the reality happens, right? So we have only 72 subjects, which is not a lot, but we have so many data, and it was more than enough for the PhD project. And we tried to put it in like stepwise linear model and other like classification model, but it was not actually good approach given our like number of subjects. So we thought we will do it as exploratory approach. So as I summarized there, we have like 5 or 6 areas that all the results are kind of coming down together, like cerebellum or like parahippocampal or like the occipital cortex. So we have some specific regions. We are trying to put them together to explain why there is less blood flow, why the blood oxygenation, like oxygen is like extraction is less, why there is more susceptibility. We are trying to analyze the effects of these findings in physiologically, and we can say, okay, concussion is affecting the brain in this way. And I would say this will help like the future studies, like already there are a lot larger studies, like with ICRP, they have like hundreds of subjects, and there's this NFL study coming up, they will have like thousands of subjects. So as I said, this is kind of a preliminary study for those that will help to direct the research process in those studies. Thanks so much.
Yeah, I'm excited to hear where your research is going and where the project is leading to. And another question I have for you is, currently, how is concussion diagnosed?
Okay, so this is also a good question, because currently the medical imaging, like you can if you have a concussion, you cannot get MRI or CT to diagnose you. First of all, those are expensive, and secondly, they cannot diagnose concussion, the clinically available MRI or CT, because they don't know what to look for. If you have a like more injury, like severe injury, you have hemorrhage, you can see it in the medical imaging. But the concussion, you have so many symptoms. You have dizziness, you have nausea, you have headache, but you don't know what's happening, and you cannot see from the clinical imaging. So currently, our only hope is the medical questionnaire. So there are different questions, like for example, the sport concussion, they have SCAT scores, like SCAT 3 or SCAT 5. So they have like, will ask you like, how is your dizziness, 1 to 10? How is your headache, 1 to 10? Are you feeling nausea? Like we have these questionnaires, and like there's balance test, all these things, and there are physicians who will look at it and give you an idea of, okay, so you are concussed, maybe take a rest. Some are now suggesting maybe do some exercise. So all these things, but I'd say we are still at the very preliminary stage in terms of that, because we cannot objectively diagnose concussion with imaging.
Thank you. If anybody else has a question they want to ask, feel free to post it in the chat. Otherwise, I do have one last question for you. So in terms of recovering from concussion, like what are the different factors that can contribute to like who can recover fast versus slow?
So it can depend on different factors. For example, like history of concussion. Did you have a previous history? Because sometimes we see people who had previous concussion might need more time. And there are other factors as well, like for example, male and female, they might recover differently. Like there are some evidence of that, and that's another research question. So that's what we're trying to understand, like what actually contributes to the recovery process, and we're trying to understand it from the biological perspective of the brain itself, like how the brain is affected, and what effects cause quick recovery and what effects cause like delayed recovery. So for all these subjects, we also have the recovery time. So how many days they took to recover, and we're trying to find the correlation between that and our like imaging findings to see like if we can explain recovery from that.
Okay. That was a great answer. Thank you very much. Well, those are all the questions we have for today. Thank you to all our presenters for sharing your research with us, and for all of our attendees for taking the time out of your week to join us and pose questions for the Q&A. It was a pleasure to have everyone here. Feel free to connect with the Graduate College on social media and visit our website for information about our upcoming events. And with that, we hope to see you for future events. And thank you everyone for joining us.